Welcome to Physiological Psychology. We're going to be talking about the synapse. Let's first look at some of our information from the neuron and see how well we remember things. If I ask you, these cells are responsible for myelination, the white matter in the brain. Which is it? Are these neurons, astrocytes, oligodendrocytes, or microglia? And the answer is the oligodendrocytes. This type of neuron is present in the cerebellum. These neurons have a large number of dendritic branches in a single plane, which means they're fairly flat. Are these pyramidal cells, Purkinje cells, monopolar cells, or retinal ganglion cells? And the answer is these are Purkinje cells, very special to the cerebellum. Opening these channels causes the neuron to depolarize. This is sodium, potassium, chloride, or calcium, which are the associated channels. And the answer is sodium. Sodium channels, when they open, they allow positive sodium ions to rush into the neuron because sodium has two gradients working for it. At rest, there is much more sodium outside than inside, and at rest, it is much more positive outside than inside. This means as soon as you allow sodium the opportunity to come in, it wants to come in, bringing that positive charge with it, which then diffuses, opens other voltage-gated sodium channels, and allowing more and more sodium to rush into the neuron, depolarizing and sending the signal down along the axon. Well, neurons communicate in two ways. They communicate through electrical and through chemical signals. And when we talk about the neuron, specifically, we talk about electrical communication. It is through chemical communication that the synapse works. So you travel quickly. We saw that neurons can fire along the axon up to over 200 miles per hour in terms of how fast that signal is sent. However, the speed of transmission of the axon, at the end of the axon, where it is separated from a dendrite from another neuron by the synapse, is relatively slow. It's a chemical diffusion, right, rather than an electrical signal. So we're going to be going over what this synapse looks like and how neurotransmitters work. Well, neurons communicate by transmitting chemicals at these synapses. So this term was only coined in the late 1800s when they discovered that there was this space. And the space was figured out by guessing. They weren't really able to, to investigate the properties of it, right? We said Ramani Cajal was able to see that these neurons really were separate, not growing into each other. But the synapse is microscopic and very difficult to see, even with very powerful microscopes. So Sherrington and others investigated this. And what's interesting, too, is that neurons didn't have a name until 1891. There was no term for this. And what we're looking at here is the reflex arc. So here's a bigger image of this reflex arc. Think about it in terms of this. You reach out and you touch something that's hot and you pull your hand away. You do this so fast that you're not aware that you touched something hot, right? That actually takes time. In fact, the pain signal takes time to get there. You will often be aware that you burned yourself before you feel the actual pain, because the pain signal takes a while to get there and to produce all the other factors associated with pain. The reflex is spinal, which means you touch something, it goes to the spinal cord, which is kind of reversed compared to the brain. In the brain, the gray matter is on the outside, the white matter is the tracks connecting the inside. In the spinal cord, it's the white on the outside and the gray on the inside. And inside is where you have that processing, that higher level processing, where signal, something's wrong, pull your hand away, right? That then is sent to the brain for further processing. So that's how the reflex arc works. Now, it's not automatic, and the reason for that is the time it takes to pass between synapses. which means it's not instantaneous, right? This is a process that happens without brain input, but it takes a little bit of time anyways. So you have this sensory neuron from touching. You have this interneuron in the spinal cord that's basically then sending a signal to the motor neuron that says pull away. So how did Sherrington go about figuring this out? Well, part of the clue is we are able to measure the speed of transmission at the neuron. If you apply an electrical stimulation to part of your body will respond. You can make your arm twitch, right, by applying an electrical signal because neurons are communicating through electricity. Really interesting recent investigations into spinal cord injuries involve bridging that space that's broken, that's not functioning, through electrical stimulation, right? Sending an electrical stimulation in order to cause movement. However, our bodies are extremely precise, and doing this properly is quite difficult. So Sherrington is measuring the speed of conduction along an axon. And then he measures how far is the distance of a reflex arc. You can measure, how long is a neuron from touching to the spinal cord and then back out to the motor, and compare that to the speed of just the transmission in one direction. Well, what he found was that reflexes were slow by comparison, right? So it wasn't the same speed as simply conduction along an axon. Something was slowing it down. And again, we're talking about milliseconds. We're talking about a very fast process, but that it was still measurable. And he noticed that there must be something happening here that's slowing the process down. It can't be all electrical. And part of his, observation that was so interesting was that you also have inhibition, all right? And you also have this summation. So you can have multiple signals coming into the same neuron to tell it to do things. You can also have a neuron that when it's doing something, it's inhibiting something else. And we think about that in terms of, in a way, protecting yourself. So let's take a look at the synapse, and we're going to unpack this. So the terms that I think are important to note here are, you've got the vesicle. Now, at the top of the image, you have the end of an axon. This is a terminal button or an axon terminal, right? But we just want to remember this is the axon. Another way it's also described as presynaptic. So the axon is presynaptic because it's before the synapse. So I think it helps to visualize the synapse in this way. The axon here, right? Then the space of the synapse, and then the dendrite here. That's the simplest way of thinking about it. So this, the part labeled B is the dendrite that is getting the information from the synapse. So A is releasing neurotransmitters, and B is having them attach to receptors. So the neurotransmitters are in these vesicles. Now, the synapse, at the presynaptic part, at the axon, you also have autoreceptors. So the synapse, the axon, is constantly monitoring how much neurotransmitter is in there, right? How much is being dumped. And so how much is kind of being brought back inside. You have this other part on the other side, the reuptake channel. So the autoreceptor is basically saying, hey, how much should we be taking in? Is there too much here? Should we be breaking it down? Should we slow production? There's a lot of factors involved. You are, no matter what anybody says, incredibly well balanced because you have in your billions of neurons these synapses that are constantly making sure that the neurotransmitter balances are about what they should be. Now they're able to change. They are absolutely able to change in order to store new information. But generally, they maintain this fairly equilibrium. In the same way, your sodium and potassium levels outside of your neurons and inside of your neurons are being balanced to allow the neurons to fire properly. Well, we also have these calcium channels, and it is the calcium that's entering the cell at the synapse that allows the neurotransmitters to be released. And we also see what's called exocytosis. Exocytosis is the process by which inside the presynaptic neuron, inside that axon, you have these vesicles filled with neurotransmitters. And when these neurotransmitters, it's time to release them, they merge with the cell wall and open, dumping out the neurotransmitters into the synapse. Now the dendrite on the other side, this is B, this is postsynaptic, right, after the synapse. And what we see here are all of these channels. You have these ion channels, and you have these receptors. And sometimes you can have a receptor that's connected directly to an ion channel. This is really quite common. And the receptor will attach and open the ion channel. And boom, sodium comes in. Sodium starts to depolarize the neuron. You can also have other effects too. You could block sodium channels and say, hey, don't fire. Okay? So you have a lot of these processes happening that could be affecting the speed by which the neuron is communicating. And also we recall in the brain, you often have multiple competing messages. And it's not about whether you fire or not, it's about how fast you do. Because every neuron has a base rate of firing. Every neuron is going to fire a certain number of times per minute on its own, on average. And changing that is going to change something about processing, perception, etc. Now here's another image that's a little more what it looks like, right? A little more specific, where you get kind of a better idea of how far apart those things are, how far apart the presynaptic and postsynaptic membranes are. The presynaptic membrane being the very end of that axon, the postsynaptic membrane being the very beginning of that dendrite. And we see the same kind of things with your vesicles filled with neurotransmitters. We see that they are releasing those through exocytosis. And as these neurotransmitters attach to receptors on the postsynaptic neuron, the postsynaptic neuron does a thing. So our terminology here, the presynaptic neuron is what delivers the transmission through the release of neurotransmitters. The postsynaptic neuron is receiving that transmission through the attachment of these neurotransmitters to receptors on the postsynaptic neuron. And here's the beginning. We're going to see two types of these things, EPSPs and IPSP. And EPSP is an excitatory postsynaptic potential. Basically, an EPSP is going to do something to depolarize the neuron. So these signals that come from the presynaptic neuron tell the postsynaptic neuron to do something like open a sodium channel and depolarize. Increase your firing rate. Now, what's important here is just because a sodium channel opens, it doesn't mean the neuron's going to fire. It doesn't mean you're going to do much. So what you end up with is Sherrington's idea of summation, and in this case, temporal summation, temporal being time. The idea is, I might send, this first neuron might send a signal that says fire, but it's a little one. It's not enough to make it fire, because remember, here's my neuron, and here, if I consider my hand, here's the cell body, and these are dendrites that all have one synapse. I might have 5 synapses here that are constantly firing and saying different things. One might be saying fire more, fire less, basically excite, inhibit, depolarize, polarize. And so the neuron is getting these mixed messages. Well, what happens if you get a message that is not enough to make the neuron fire, but it depolarizes a little bit? Well, over time, what we see here is these little boosts are going to decay. So you open a sodium channel, the signal goes away, and you close it, and those sodium ions diffuse, sodium-potassium pump dumps them out, and your neuron returns to its resting state. That's what we're looking at here on these little graphs, right? So at the very top, we see the same kind of thing. You have an electrode stimulating a neuron, right? At the very bottom, we see each of these pulses is the same. You get one pulse, it decays. But now you get a pulse, it decays a little, you get another pulse, it decays a little, you get another pulse right away, and it's enough to go to threshold, and then boom, the neuron fires. Temporal summation is adding up over time. And so you can get a lot of little signals that aren't enough to make the neuron depolarize, and then it depolarizes. Now, what's interesting is that Sherrington did this in late 1800s technology. They weren't the same kind of thing. He was really doing very subtle things to see how could he how could he cause a reflex in a dog leg, because we're looking at the reflex, the involuntary. When you had the reflex, when you were little and you went to the pediatrician and they hit you on the knee with the hammer, right? Well, did you try to kick really good to show oh, look, it worked, or try not to kick? None of that mattered. What they were looking at is that muscle twitch, right? So whether or not you kicked, it didn't matter. What mattered was the reflex, because that's a reflex. And so what that's telling the pediatrician is this a typical reflex, or is it slow? And thereby being something wrong. The other type of summation Sherrington figured out was spatial. And the idea here is, what happens if, okay, you've got this neuron with, these different dendrites, all these dendrites, and you got, let's say, 3 signals coming in. Each of those signals from other neurons is not enough to depolarize you, not enough to make this neuron fire. But these 3 little signals, all at the same time, well, these are enough to make the neuron fire. So here in this image, we see 3 synapses, right? 3 axons connected directly to the neuron across the synapse, across this new neuron. Each of these signals is small, not enough to make the neuron fire. But when it gets them all at the same time, boom, it's enough to cause depolarization. And here is EPSPs and IPSPs together. The idea being, over time, now this is key. Over time, here's your nice and flat, resting potential, right? You're just kind of going along at resting potential, and you get a signal that says polarize or depolarize, polarize, depolarize, polarize. And when you get enough depolarizes together, that's when the neuron hits threshold and fires. That's the idea, right? You get enough of this signal to depolarize, you depolarize. But what you have is this symphony of incoming information, not even a symphony, a cacophony, noise. So much going on that some of these signals are saying fire, some are saying don't fire, polarize, depolarize, all these different messages. And you reach threshold and the neuron depolarizes. So again, EPSPs, excitatory postsynaptic potential. And your IPSPs are inhibitory postsynaptic potentials. And this is one of the genius of understanding neurons. I think one of the hardest things to really kind of wrap our heads around, which is some signals are excitatory and some signals are inhibitory. When we talk about incoming signals to neurons, we think of a signal input or a few inputs. But there's so many of these things. And when it comes to things like reflexes, maybe it's only one signal, right? It's like, hey, this is hot, get your hand out of there, and it has to happen fast. But when you think about something like memory, trying to pull something out of your memory, or trying to picture a sunny day at the beach under the palm trees with a cool breeze and the smell of salt in the air and all these things, there are so many neurons involved and so much signal happening that there's these contradictory messages, right? You're thinking of that sunny day, you want to inhibit the thought of clouds. Now, Sherrington noticed the inhibitory part, the inhibitory postsynaptic potential, by looking at when he caused the reflex in one dog's leg, the other legs remained extended. And not only that, they were very difficult. The dog couldn't bend them. It was hard to bend them at the time. Which meant what happened was, when he caused that reflex, those other 3 legs were basically getting the signal that says, don't bend. Because look, if you were to stand on one foot, you stand up, you bend your knee, you stand on one foot. If you were to bend the other knee at the same time, you'd fall down, right? Same thing with a reflex. You pull your hand away, you want to hold on. You don't want to do the same thing at the same time. And for a quadruped, for an animal with 4 legs, same kind of thing. You need these things to be coordinated. And so, moving one doesn't the other. Now, we can see this too. right? Flex your bicep and your tricep at the same time. Get them as tight as you possibly can. And you can't get them both at the same level as you would if you did them independently. The reason is when you bend your arm, you're basically sending a signal, right? You're using your bicep to bend your arm. You're sending a signal to your tricep that says, relax. So if you were to use both of these muscles at their max at the same time, you could cause damage, right? Rip the muscle right off the tendon. When you extend your arm, right, now your bicep is relaxing. So you're basically getting these signals where they're saying, do this, don't do this. It's protective. It's part of this process. Let's look at the configuration of a reflex arc. What is the correct configuration? We've got sensory, interneuron, and motor neurons involved. What is the way that this works? Well, what happens first, right? Is it sensory, interneuron, or motor? And what happens last? And is there an in-between? And the answer is the sensory is first. In the reflex, you have to sense it. The interneuron is the processing in the spinal cord itself. And the motor neuron is the movement, the reflex, the moving or pulling away. These make a neuron more likely to fire. So what have we talked about? Neurotransmitter breakdowns, sodium channel blockers, IPSPs, or EPSPs. And the answer is EPSPs, excitatory postsynaptic potentials, make a neuron more likely to fire. Of course, you have neurotransmitters, but if you break them down, you're doing different things at the synapse. Sometimes it may make a neuron more likely to fire, but we need more information to answer that question for certain. Sodium channel blockers, we know, make a neuron less likely to fire. IPSPs are polarizing, right? They make the neuron less likely to fire. So EPSPs, even if they don't cause an action potential, they make the neuron more likely to fire. Now let's consider a scenario. A presynaptic neuron, right, information coming, sends an EPSP to the postsynaptic neuron. It does this repeatedly, right? So it's like boom, 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 sending the signal. Each signal is not enough to make the neuron fire, but repeated stimulation makes it fire. What are these called? What is this called? Is it inhibition and excitation? Is it spatial summation, temporal summation, or IPSPs? And the answer is this would be temporal summation. Repeated stimulation over time causing a neuron to fire. Now what's interesting is neurons all over your brain have different natural firing rates. They're spontaneous firing rates. Your Purkinje cells in your cerebellum, right, the majority of the cells in your brain, like 70 billion cells, lots of Purkinje cells among those cells, they fire at 40 to 50 hertz. Now hertz is something, we'll talk about it when we talk about hearing, but we're also talking about it in terms of things that happen rapidly are often measured in hertz. Your computer screen is not a static image. It's refreshing at about 60 hertz. If you've ever seen a video of something like a helicopter, where the helicopter appears to be moving, the blade's not, it's because the blade is spinning at the same rate of the recording, right? So it's just kind of lining up perfectly, and it's cutting out all the movement. So all you see is this static blade, right, happening many times per second. Well, Purkinje cells at 40 to 50 hertz are firing 40 to 50 times per second. The inputs to the Purkinje cells are firing about once per second. So these neurons are slower, your parallel fibers and such. Homeostasis neurons, these are the neurons in your hypothalamus. So your hypothalamus is heavily involved in homeostasis, and homeostasis is maintaining your body's life processes, basically. And this will be in much more depth when we talk about hormones that are part of this process. But the idea is you have a lot of processes happening in your hypothalamus that maintains your blood pressure, your body temperature, etc., within a very narrow set of ranges. And so these are firing at less than 10 times per second. And there's a group of cells in your hypothalamus called SCN cells that are involved in telling you is it daytime or nighttime. They're getting input from light. So if it's daytime, they fire 7 times a second. If it's nighttime, they fire 2 times a second. And their outputs know that the speed of firing says produce certain hormones or other hormones. So this pace of firing influences hormone production, and that's how those work. Hippocampus CA3. Your hippocampus area, CA3, has your major outputs to your thalamus. It's heavily involved in memory formation, and they fire less than 1 per second, 0.2 to 0.4, which is once every 5 seconds to once every 2.5 seconds. These are slow. This is their spontaneous firing rate. But for all of these neurons, inputs can change that rate, speed them up or slow them down. And that's the idea. Your brain is constantly adapting the firing rate. Your neurons are firing at different rates. Neurotransmitters travel across the synapse. We know this. Well, the discovery of these things and how that they worked from a chemical standpoint was discovered in 1921, another Nobel Prize winning discovery. And what Lowy did was he was working with other researchers and kind of got this idea that certain medications were having cross purposes. Drugs that speed up and slow down were kind of canceling each other out. And so he's wondering why this was and what was happening. So he took a frog heart and he stimulated it so that it beat so that it beat slower, so that the heart rate slowed down. And this is called, you can actually do this through vagal nerve massage in like a living organism to kind of help relaxation, biofeedback types of things, and slow the heart rate. Well, around the heart was a solution, right? It's in, a solution keeping it alive. And so he took a little of that solution and put it in a second heart, on a second frog heart. And that heart also slowed down. Now we think, what's going on? Why is this liquid outside of this first heart causing the other heart to slow down? And the answer is that when that first heart slowed down, what made it slow down was the electrical stimulation, but then to communicate within it was to produce chemicals, neurotransmitters that said, slow down. So taking some of that liquid with those neurotransmitters in it and putting it on the new heart slowed it down. Now, I don't know if Lowy figured this out in a snap or it took a long time to think about this and go through the logic, but whatever it was, he figured it out and he was able to do this. Some neurotransmitters are synthesized in the brain. So we want to look at where these things come from. And some of these are going to have to be gotten from other things, right? So some are being synthesized, some are in the diet. And so you have your action potential traveling down the axon, causing the release of neurotransmitters. They cross the synapse attached to receptors, and then they separate. So they're not staying on those receptors. They disconnect, and then there's 3 main processes that happen. They can either be broken down by things that are in the synapse, and this is going to be key for later. There are molecules, there are enzymes in the synapse that will cause the breakdown of neurotransmitters. So you get too many, cool. You maintain the equilibrium, you maintain the balance by breaking them down. You can also have them diffuse away, right? So they'll just kind of like diffuse and kind of float away. You can also bring them back up into the presynaptic neuron. So reabsorb them into the presynaptic neuron. The major categories of neurotransmitters are, and the ones that we were going to care about, amino acids. The number 1 neurotransmitters used in the brain are amino acids. And that should make sense. If the evolutionary process is putting pressure and creating these neurons, and you've got synapses, and you want communication, you're going to use something that's there, right? That's commonly there. Other ones take more, are more complex, have a process involved to balance them more clearly. And I'll go through a number of these and kind of go through. So let's look at specifically the first set, your amino acids. And the 2 main ones here are glutamate and GABA. And these are excitatory and inhibitory. Glutamate is your brain's primary excitatory neurotransmitter, and GABA is your primary inhibitory neurotransmitter. And the synapses, there's two main types of synapses, and the synapses that work with these two tend to be what are called ionotropic. The idea is you release GABA and you're going to stop these from firing. You're going to slow the firing rate. You release glutamate and you're going to do things like, boom, open an ion channel right now. Open up a sodium channel, depolarize. So those are the most common. Another common one, the number one used in movement. So acetylcholine is causing movement, muscles to contract and things. It's a primary neurotransmitter there. It's a modified amino acid. So it's like an amino acid, but it's got a little something else on it. And we see in the image here some of these receptors. Nicotine is, it affects acetylcholine receptors. And so what happens is you have got these receptors called nicotinic receptors, right? And they're named, a lot of receptors are named after the drug that they figured out, oh, this is what works. In the same way that endorphins are named after drugs. So here we have nicotine, where it causes an effect and eventually kind of causes this effect that releases dopamine. Nicotine, smoking, right, cigarettes and associated products causes a release of dopamine that causes a pleasurable experience, even though it takes a lot of work to get past that for most people. That initial, this sucks. It seems like it takes a lot of work to start smoking, and yet marketing makes us do it. Seems so glamorous. Those 1930s Hollywood stars always holding their favorite brand. Well, then we have the monoamines. This is the biggest class in terms of targeted things for mood. And these are probably the ones that are most common for you, right? So you got two main classes here, the indoleamines and the catecholamines. And serotonin is the major indoleamine. There's others, but we only care about this one. SSRIs, drugs that affect mood, are going to work in this way. Right? Drugs that affect mood are going to work on your serotonin synapses. And we see here some interesting things that are happening. SSRIs are selective serotonin reuptake inhibitors. What this means is selective, older antidepressants, or I'm not even in love with that term, older drugs that were given for depression would affect all of these monoamines: serotonin, dopamine, norepinephrine, and epinephrine, tricyclic drugs, and MAOIs, right? MAOIs, right? Monoamine oxidase inhibitors. Monoamines, MAO. Monoamine oxidase is an enzyme that breaks down monoamines. So you have monoamine oxidase in your monoamine synapses. You get too much serotonin, dopamine, norepinephrine, and epinephrine in there, and you start to break it down, again, to maintain the balance. MAOIs stop the function of MAO, meaning that in your synapse you have more serotonin, dopamine, norepinephrine, epinephrine. The problem is there are more side effects for these drugs. So the early drugs given for depression had more side effects. Well, SSRIs had fewer because they only worked on the serotonin receptors. What would happen then is, now these didn't work like MAOIs, they didn't stop the monoamines. So your monoamine inhibitors, your monoamine oxidase is still floating around and they're breaking down the excess serotonin. But your brain, your neurons, right, that normally would take serotonin back up into the presynaptic neuron. So you release serotonin, there's so much in there, and it comes back to be recycled. Would stop taking it back in, which means you would have more available. And if you have more available, you have more, serotonin you're working with, and the idea that it contributes to, mood is the reason that these drugs were, created and marketed and used for depression. So that's the idea of how SSRIs, right? So to kind of look at it, serotonin is an SSRI, drugs given to treat depression are often SSRIs that affect serotonin. selective serotonin reuptake inhibitors, which means serotonin is not being pulled back in. Now, here's the issue. Long-term use, and most studies, when you're looking at efficacy, are like 3 to 6 month studies. It's only recently that research has been looking at the long-term effects of taking these drugs. And any drug given for a length of time, your brain is going to adapt. So how might your brain adapt? It might get rid of some receptors. It might produce less serotonin. It might boost the production of monoamine oxidase in order to break it down. So anytime we're affecting brain chemistry, we want to be careful. The good news is these drugs don't seem to be as harmful as things like cocaine. So let's consider drugs that people take, right? Abuse type drugs. And we look at cocaine, and what we see in this image here is cocaine works in a similar way. It stops the reuptake. So you release dopamine and there's more dopamine available, right? So dopamine, norepinephrine, and epinephrine are all related in terms of their class and in terms of their production. So you end up with these drugs, these neurotransmitters that are similar. So when we see things like drugs that people take, we think about ways that they're going to boost the availability. And the way they do that is, in this case, they stop the reuptake, right? Well, you look at things like food. Dopamine increases in response to pleasurable foods. What are your favorite foods? If you're eating chocolate or something that you just love, it creates this dopamine reward. Cocaine creates this massive dopamine reward, and so you end up with increased dopamine. In drugs of abuse, however, you tend to see a much quicker brain adaptation, which means if someone's using cocaine a lot, or amphetamines, which have a similar mechanism, your brain is going to produce less dopamine and be less responsive to dopamine, and that is how dependence works. Your brain then needs more than it did before. It needs to boost those levels in order to kind of go back to that normal level of function. We look at other neurotransmitters. Endorphins, I mentioned, are named after drugs. That sounds weird, but the way that we discovered these neurotransmitters came from, we already knew, from prehistory, people were taking drugs and mind-altering stuff, right? So you had people brewing alcohol as soon as they figured out that they could. And you had people using things like opium, which was very easily derived from the opium poppy. Well, opium contains morphine, and endorphin gets the name from endogenous morphine. It was easy to see what morphine was doing to the brain, more easy to see that than to see what the neurotransmitters themselves. These things are tiny and very hard to track. And research on it is difficult because you can't look at a neuron in a living brain and see how much dopamine is being produced, how much serotonin, if it's a GABA, whatever's being involved there. So a lot of this is kind of guessing. In the same way that people like Sherrington and Lowy were able to deduce the function of neurons and synapses and neurotransmitters without being able to see them. So that's the idea. The more that we understand about these things, the more we start to put pieces together and see how they work. So if any of this is unclear, it takes time. It takes time. There's no way those Nobel Prize winners figured this out by reading a textbook one time. Or looking at some definitions. Understanding these complex processes, like how a neuron fires, takes work. Other neuropeptide that we care about is substance P. It's involved in pain. It's the pain signal. Purines, like ATP, we know what that one is because that's the energy, like the batteries in the cell, right? You convert glucose into these ATP molecules, and every time you do something, you use ATP. So when we look at drugs in the brain, we're saying, okay, how do these things work? Well, you can change production. You can increase or decrease production of neurotransmitters. You can change release. You can make it hard for a neurotransmitter to be released. You can block your ability for them to attach, and that's not even here, right? So you can basically have these neurotransmitters be released, but there's nothing they can attach to. That's common in drugs given for psychosis. So drugs for people who have maybe schizophrenic type symptoms. They might have block the ability of dopamine to do its job. So dopamine's still floating there, but it can't connect to its receptors. You can change reuptake. You can make it, like SSRIs, which are blocking the ability of serotonin to come back up. You can change breakdown, like MAOIs, that make it hard for monoamine oxidase to work. So you've got more serotonin, dopamine, norepinephrine, and epinephrine floating around in the synapse. You can also mimic. So you mimic drugs like morphine. Morphine mimics the effect of natural endorphins. So it does the same kind of thing. So there's all these different ways, but drugs in the brain, drugs that have a psychiatric, psychological, neurological effect in the brain, have to be able to cross the blood-brain barrier, and they have to be able to do something to change function in the brain in regards to neurotransmitters. And a lot of this is still unclear, especially in terms of drugs for mood. So it's a really cool area of investigation. You've got two terms here involved in neurotransmitter use, and that's agonist and antagonist. Agonists make the neurotransmitters more effective. It basically increases the effectiveness of the neurotransmitter. And antagonists decrease the effectiveness. Now, how do you do that? Well, an agonist could make more of the neurotransmitter, or release more, or block the reuptake, or decrease the breakdown. The antagonist could decrease production, or block the function at the postsynaptic membrane. So there's all these different processes that are happening. And remember, this is happening throughout the brain at trillions of synapses simultaneously. I mentioned ionotropic receptors. So there's two main types of receptors, two types of receptors at the postsynaptic neuron. So you release your neurons, you detect them, right, at the dendrite. And your ionotropic receptors are fast. This says, boom, open a channel, right? GABA and glutamate tend to be this. Their direct effect on an ion channel. Metabotropic receptors are slower, but they can cause longer lasting changes. So we go back to, if you recall from discussions of the neuron. In the neuron, you're going to be changing the neuron itself, adding channels, deleting channels, changing receptors, etc. Growing synapses, growing the dendrites or axon terminals, boosting neurotransmitter production. What causes this? And the answer is generally metabotropic receptors. These are sensitive to change, right? And so if you get a lot of stimulation, you may cause something. You may cause a multi-step process to open an ion channel. You may also start a multi-step process that then goes to the nucleus, opens up, right, causes the histone to release a strand of DNA, copies it, and then goes and creates a protein. So these receptors are much more interesting. Ionotropic, boom, open an ion channel. Metabotropic, do something that takes longer and may cause permanent change. And so you end up with, you could still have a metabolic receptor that opens an ion channel, but you also have all these other options available. As noted, neurotransmitters some are produced in the brain, some require external things. So for instance, acetylcholine requires choline, which is found in milk, eggs, and nuts. Tryptophan is a precursor for serotonin. And things like catecholamines have an amine and a catechol group, and a process where they can actually be adapted to become each other, right? Dopamine and norepinephrine and epinephrine are all part of this process. So we're constantly synthesizing, and again, a normal diet isn't going to have any deficits. So how are we with these things? Which of these is a monoamine? GABA, acetylcholine, nitric oxide, or serotonin? And the answer is serotonin. and I cannot overstate the importance of knowing the monoamines: serotonin, dopamine, epinephrine, and norepinephrine. And interestingly, epinephrine is also called adrenaline, and norepinephrine is also called noradrenaline. Which of these is an amino acid? GABA, acetylcholine, nitric oxide, or serotonin? And the answer is GABA. The two main neurotransmitters in the brain, GABA and glutamate. Well, thank you, and